Welcome to the Love Your Life and Yourself podcast, the weekly show where science meets soul and mom chaos meets mind mastery. You're not just a mom. You're a woman with dreams, a career, a body that needs care, and a soul that craves more. This podcast is your permission slip to stop surviving and start thriving in motherhood, health, career, and stress resilience. I'm your host, Dr. Ashley Rath, and every week we bring you actionable insights and transformative conversations to help you create a life you truly love. From evidence-based wellness strategies to authentic self-discovery, we'll explore what it takes to thrive through expert interviews and real unfiltered stories. Let's dive into today's episode. All right, welcome back, everyone. I am so excited for today's guest. I have been sharing his research in my keynotes for the last 10 years, and now I get to talk to him in person. We have Dr. Randy Jirtle. He has pioneered epigenetics and the research behind it, and really known for how environmental factors, especially in early development, can alter this epigenome and influence really kind of adult disease. So right now, you just retire, right, Randy? So
No,
let's
I was at Duke for 35 years. I joined the faculty at Duke in 1977. I retired from Duke after 35 years there in 2012, and I thought I would be retired, but then my group went over to NC State, and Catherine Hoyo, who was in my laboratory at that time, not physically, but worked directly with us, went over to NC State and asked me to help her set up an environmental epigenomics human disease susceptibility program there. So I never really had a retirement, but I don't have to take care of the university things anymore like I did when I was a actual faculty member. Now I'm just an adjunct faculty member over at North Carolina State since then. So I've never actually really retired, but don't have as many responsibilities as I did when I was truly a full professor at Duke.
Fair enough, which is probably a good thing. You get to, obviously that means you love what you do when you can't just fully retire from it and you still got your feet a little bit wet. So what I would like to know is your just journey. So from beginning to end, from what I have found out, you went from nuclear engineering to epigenetics and imprinting. So tell us about your whole career journey and really how you got to where you are now.
Well, I did my undergraduate work and graduated from the University of Wisconsin-Madison in nuclear engineering, and I was actually, what do you call it, given the right, in effect, to run the reactor at the University of Wisconsin that was from the AEC, the Atomic Energy Commission back then. But during that period when I was in nuclear engineering, I had a series of lectures on the biological effects of ionizing radiation on humans and biological materials rather than just physics. And I was really captured by that. And ultimately, I went from nuclear engineering into radiation biology. So that's how I got into biology, but with a very heavy quantitative approach to biology and science compared to most biologists at that time and maybe even now. And that's probably why I was able to do the type of work that we'll talk about later on. The human imprintome and stuff like that is very mathematical, bioinformatic type research. And that all started back then. And then when I came down here, I was working more in cancer research. And at the time, we were looking at a gene that was called the IGF-2 receptor and identified it as being a tumor suppressor gene. And that was the first gene that was identified to be genomically imprinted. And what this means is that even though with imprinted genes, you inherit one copy from mom and one from dad, one of the copies is turned off epigenetically, so you only have one copy that functions. So those all become vulnerability loci or regions in the genome because only one copy works, even though you have inherited two. So it's like you're flying in an airplane and you have an engine on both wings, but before you take off, Mother Nature has turned off the gas to one of the engines.
Hmm.
And you can take off, you can fly, and you can land with a single engine, but it makes you feel very comfortable knowing when you're at 30,000 feet that you have both of them working. Well, we
Right.
have a set of genes in our genome in which only one copy works. And they're all revolved around metabolism and growth. So they play hugely important in development, therefore, importance in development of the brain, the body, and metabolism itself. And since you only have a single copy, now either a single mutation, because you only have one that works, and knock it out, or if you're talking about epigenetics, you can either have no copies because this one's silenced also epigenetically, and that's too little, gene expression, or you can have loss of imprinting and you have too much. So either one of these cases is bad. So you only need one. And if you go too much or too little, you have problems. So when I first found out about this, and Denise Barlow was the one that did this work, she really directly got me involved in this field of epigenetic research because it was clear to me that epigenetics was playing a major, major evolutionary role in effect, disease susceptibility ultimately. And I told the lab we found out that this was an imprinted gene, that I was taking the whole lab into the field of epigenetics and genomic imprinting. But I have to admit, this is at around 2000. Yeah, right around 2000. No, it was before that. It was 1990s, early 1990s. I didn't know how I was going to do it, but we did it. And that's how I got into the field of epigenetics. It's when you look at it from the outside, it makes no sense, but when you're living it, it makes a lot of sense because I know exactly when I came to these Ys in the road and decided to go off into the field of epigenetics right around 1995. And that's geneimprint.com, which is our website, was my son actually built this and designed it back in 1995, 98. This is before anybody had websites.
Right.
You still had dial-up. American Online
Yeah.
was
Dur,
cheap.
dur, dur, dur.
Yeah, right.
Uh,
Back that era.
uh-huh. Oh,
Yeah.
I've been there. Never built a website on that though. So there's a lot of listeners that may not have heard of the term epigenetics. And for someone who may not have heard of epigenetics and really doesn't understand it, how would you explain it in a way that the general population can understand as we move through our conversation, what truly is epigenetics?
I think the easiest way to understand it is an analogy. I think of the genome, the DNA in the cell, in our cells, to be comparable to the hardware of a computer. So the epigenome, which just means above the genome, are the programs that tell that hardware, that computer, when, where, and how to work. So in effect, every cell is a programmable computer. And the epigenome is the programs, and every cell has a different set of programs that are running in it. That's why you can have, I like Mac computers, so you can have, 260 Mac computers. There's about 260 different cell types all sitting there, but they're every cell has the same DNA because one copy comes from mom and one comes from dad. So they all have the same computer, but every computer is running a different software program. Some have, Safari or Chrome or Word or PowerPoint or whatever. They're all running different computers, different programs, and they're doing different jobs. And that's in effect what every cell type is doing. And that's why we can have a single cell come into being and making 260 different cell types that are doing completely different things. It's because of the programming. So you have now the potential of the environmental factors, nasty stuff, including one of them at high doses anyway, ionizing radiation, chemicals, all those kinds of things, even food some of it is toxic. And so you can not only cause mutations in that computer, which would be in the computer world would be looked at as being a hard drive that didn't work, something physical that didn't work, or you can have a problem in the software. And as most of us know, which we almost had this morning, Ashley, there's often glitches. Not in our hardware. Our hardware was working fine. It's the software often that gets messed up and doesn't communicate properly. And all of a
Mhm.
sudden, you just got a darn disaster. And the same thing happens in our cells. You can have mutations, which is what most people in the past have focused on. And you can have what I call other people too, epimutations, changes in the programs that also cause disease susceptibility. So that's the way I look at it. And it's very simple I think, to think about it that way. Hardware is DNA,
Mhm.
software is the epigenome, and you have to have both of them working well in order to have a healthy body.
Yeah, that makes sense to me. I've actually never been explained to it like that, but I love the analogy.
It works
And
though, doesn't it?
it does. It absolutely works. Why do you think I get so excited about epigenetics because I'm not quite as nerdy as you, but I like to nerd out a little bit. And but why should the average person really care about epigenetics? Like when we're talking about it, it's like, oh yeah, we have this mutation or an epi I forget what you called it, the epi mutation.
Epimutation. It's just a
Um,
change in the epigenome, basically.
right. So why I understand it from a health benefits of like, okay, we don't if my mom has this disease heart disease or diabetes, or my dad has, this disease or a chronic illness, I interpret it as, well, with epigenetics, if I can alter my environment, eat healthy, exercise, mental, emotional thoughts, etc., we can have an effect on that epigenetics, so the groups that control that DNA makeup. But why would the average person, in your explanation, care about epigenetics?
Well, it's for exact the same reason that you just said. I mean, the genetics you're born with, and so if you have mutations that give rise to eye color differences, like your eyes are blue and mine are brown, that's a mutation. And your
Mhm.
parents both had blue eyes or they were heterozygous for blue and you came out with blue eyes. I'm heterozygous actually for blue and brown. And, so both our kids could have blue or brown, but both had brown. But next generation, we had blue and brown coming out. So, I mean, this is all mutational stuff and there's not much you can do about that. And it's very deterministic. And if you have like BRCA1, BRCA2 mutations for cancer, breast cancer formation, they're scary because there's not a lot you can do about those directly. But with the epigenome, it's plastic and you have the ability to alter your destiny in effect. So what you were saying is that changes in your diet, changes in the food and stuff, and changes to the exposure to bad environmental toxicants, let's say, that you're exposed to, all of these things potentially can have bad or positive effects because they alter the expression of genes that can tend to override some of the times the negative effects that you find from mutations in the computer that we have, the DNA itself. That's where you can now have something to do personally to help you have a healthy life, and not only yourself. These epimutations, you would think that when you go through the egg and the sperm, they're all eliminated, but they're not. I mean, the data pretty strongly suggests now there's also not only inheritance of mutations in the DNA, but there's also transgenerational inheritance of epigenetic changes. So what you do not only affects your health, but potentially will also, from the standpoint of causing changes in the epigenome, can affect your offspring additionally. So you've got to be careful. You have to try to maintain your epigenome as best you can because it not only affects you, but potentially could even affect your children. And that's the downside. People like epigenetics because they say, well, I can do something about it. So it's more of the free will part of the whole equation rather than the absolute deterministic part of it. But the other side of the coin is responsibility. You inherit something, you say, well, I don't have anything to do with that. I can't do anything about it. So that's the end of the game. That's not true with the epigenome. You have a lot that you can do about it. And if you don't, you have responsibility for potentially not only affecting your health, but the health and well-being of your children and ultimate generations to come. Is that a good description?
Yes, absolutely it is.
But you understand what I'm saying? It's a very interesting thing because what you would like personally, you flip it around, brings in responsibility, and sometimes people don't like that very much.
And I think that has a lot to do like, we talk a lot on this show about empowerment and
Yeah.
self-care, and really epigenetics tells us that our genes are not our destiny. Not that we can, like, change our DNA, but essentially how that DNA is expressed. And with our choices
The
that we
other
make.
thing, Ashley,
Yeah,
is that it can have genes can be turned on. So let's say you have a mutation that's pre-malignant and things are going down that pathway. You potentially can turn on genes now that are monitoring basically what's going on in your cells and say, there's something not quite right with you. You need to die. And it's called program death, apoptosis. So you turn on that whole program through epigenetic changes and the cell disappears. Well, if it disappears and it's pre-malignant, it doesn't make any difference to you because it's gone. So it can do it that way too, from a surveillance standpoint, maybe even through immunology, where it sees something is wrong and can take out cells that are pre-malignant or even malignant. So it can have effects like that too.
that's powerful. Like,
It is.
that's
It is. It's the monitoring basically of what's going on, and you can affect that by what you do. The problem is that we don't have a good list of targets to look at, and there's no easy way to say. It's not like standing on a scale every morning. If I'm trying, which I did at one time, I kind of have to admit I wasn't so good for a while there. I was pretty heavy, and I started losing weight. But an encouragement is every morning if you get on the scale and your weight is going down, you say, "Yeah, I did a good job." So now I'll keep trying to do a good job. It's harder when you're talking about eating and changing something as nebulous as the epigenome to keep going and say, well, if I keep going, all these good things are going to happen, but there's no readout. You don't have any positive reinforcement. And that's true still, even though now with the development of the human imprintome, at least research-wise, we can start looking at these regions and asking questions. If you eat a Mediterranean diet versus the standard Western diet, does that have any effect on the methylation of these areas that we know have to be one copy needs to be methylated and one unmethylated. So you can monitor that now because we know some of the targets. Not all of them, but at least it's a place that we can now start.
Where do you see that research evolving the fastest? Is it in diet, like Mediterranean versus Western, or is it in, maybe breath work or meditation, or I don't know. I mean, there's all sorts of different things that you can do from, quote, like, biohacking or whatever you want to call it, or just healthy lifestyle. But do you see it moving in a certain direction, um, versus one or the other?
I think, and it's because we're probably going to do it ourselves, I think it's going to start in nutrition. The thing about, Mediterranean diet is pretty well set up and people kind of know what it is. You say it and fruits and vegetables and not so
Mhm.
carbs and things like this. I mean, people kind of know what it is, and we sure to heck know what a Western diet is. Lots of
Right.
don't sweets and stuff like this. I mean, we know what the calories come in from that, and you have pregnant women, and I think they will be willing to do this, to look at this, and now we can have ways and areas that we can look at easily. Actually, these imprint regulatory elements, we have an array now that we can use quite easily to assess the methylation levels there. And it's not exorbitantly expensive to do it anymore. So now you can start doing epidemiology studies. So you can ask simple questions. If you have women that, and I'm not going to do this, but somebody is going to do this, coming into the clinic, you just basically split them into two groups. As long as you try to match up, all the things that epidemiologists match up, and you have one eating this and one eating that, and then you get out at the end and you look at them and you look at your offspring, and you start seeing what the effect did that have on this class of genes, which are these imprinted genes. That's a simple question. And it'll be interesting to see what happens. My guess is we're going to see some quite interesting results, actually. So
Which
that's
is
where I
kind of like your oh, sorry, go ahead.
No, I was gonna say that's where I see it happening first, because it's clear what you're doing, people know what you're doing, and
Mhm,
depending upon what the results are, then the things about meditation, all this can exercise, all of this can affect it, and you can do the same kinds of studies.
mhm,
We have endpoints now. See, these imprint regulatory elements come in from the egg and the sperm, so that's from the previous generation, they were methylated or unmethylated. And then right after fertilization, you get a demethylation wave, so they have to be protected. So we can look at early changes, like literally right before or right after fertilization, and determine what the methylation levels is by looking at any cell in the body. And that's advantageous because now I can take a buccal swab and a blood sample. And since back at that stage, they still were an embryonic stem cell. If there's a change there, you'll see it in every tissue in your body. So if there's a problem, you'll not only see it in the saliva, but you'll see it in white cells that you take out of your arm because they come from different germ layers. And that's incredibly important in doing epidemiology because you can say, okay, now we know what happened here. This has been altered. And it's now related to some disease susceptibility that people are looking at. So that's the way you do the epidemiology studies, but you don't run into the problem of the epigenome being variable across tissues because it's not. We're looking at very early stages, and you can look at it in any cell in the body easily.
hmm,
So again, it's a
interesting.
place to start. It's not where you necessarily will end, but we've got to start in a place where I come out of engineering, in physics, you got to control variables as much as you can so you can interpret the results. So this controls it as best as we can in an epidemiology study in humans in biology.
Yeah, humans are so variable, so I can see how that can become challenging in research.
Genetically very different.
Yes. So you did something like similar to this, which this is the research that I'm most familiar with is I don't know if I'm pronouncing the species of mouse, agouti. Is that how you
Right.
say it?
Yeah.
Agouti mouse studies was groundbreaking, where essentially you guys gave these genetically um, exact same replicas, so they had the same DNA, they were cloned mice, and the mothers were giving methylated B vitamins, or half of them were given methylated B vitamins, and the other half wasn't, and their offspring and then you had tested something in between, so, like, mild doses of B vitamins or smaller doses, and then all the way up to the full dose of that methylated B vitamins, and these mice offspring of these clone genes, so exact same genetics, came out very, very different.
Mhm.
The mice that got the methylated B vitamins were healthy with brown coats, and the mothers that didn't get those methylated B vitamins were yellow and obese. And it was profound to me, and this is the study that I talk about in my keynotes, but, it's really exciting to me, to see this, to share, like, moms, number 1, while you're pregnant, you have again, it comes down to that responsibility, for the offspring of your children, to create health or non-health, but now it opens the can of worms like you're talking about for more research of, like, well, what even happens after birth, of how we can affect how our genes are expressed or visible in a healthy state. So, um What was it like? Walk me through this research. I'm sure it took quite some time, but this had to be like so exciting in a lab to be able to come up with this. And of course, you had, the discovery of the tumor gene as well. So you've had these groundbreaking moments in your career, but what did tell me about that process and what that felt like in the moment.
Well, Rob Waterland was a postdoc that did the work in my laboratory. He came out of Cornell. He was a nutritionist. So he was very, very interested in how nutrition affected health, basically. And he initially wanted to look at imprint regulatory elements. And I said, Rob, we can do that, which is, if you think about it, that's what we're doing. That experiment that you just described in the Agouti mouse is exactly the same thing that we're going to be doing in humans. But we have one now with one diet versus another diet, and we have endpoints, and it's very controlled so that you can actually potentially get some information that people can understand and maybe
Mhm.
believe from an experiment like that. So the Agouti mouse is not only were the coat colors different, but what you didn't say is the yellow animals become obese, get diabetic, and have high incidence of cancer, whereas the brown animal is healthy all of its life. So by simply changing, we're talking folic acid, which our bread has folic acid in it, vitamin B12, betaine, and choline, I think were the four. All the methyl groups, which your audience doesn't know, but the marking system, it's not like ones and zeros like in a computer for programming. In biology, we program with methyl groups. So methyl group is a carbon with 3 hydrogens, and it can bind directly onto cytosine, which is one of the nucleic acids in the DNA. And it also can bind to the nucleosome tails that the DNA is wrapped around. And together, usually when it's methylated, it condenses the DNA and the gene is turned off. And if it's not methylated, it's opened up and it can be expressed. So that's what happens in this Agouti mouse system. You're altering the methylation of a little bit of DNA, or actually a viral element that's upstream of the Agouti gene in this very specific strain of animals. So the yellow animal, that region doesn't have any methyl groups, and the brown animal, that region of DNA has tons of methylation groups. And as a consequence, the Agouti gene is regulated differently. In the yellow animal, it's inappropriately being expressed off that region of the virus, and it's expressed everywhere in the body all the time. And so the Agouti protein in the brain now binds to the melanocortin 4 receptor, which is the satiation center gene that's involved in knowing whether you're full or whether you're hungry. So it never realizes, because it's blocked inappropriately, because that Agouti gene is on all the time and blocks it. It can't sense whether it's full. So the
Hmm.
animal literally eats itself into obesity, diabetes, and cancer. In the
Hmm.
animal that's brown, where that area is shut off and goes back to normal regulation of the Agouti gene, perfectly fine for the rest of his life and dies of old age. So the important part of that study is we not only show that it shifted the coat color in the offspring towards brown, but it did it through alterations in the epigenome, not through mutations. So
I'm
disease
curious. Oh,
susceptibility in adulthood is due to what the animal, the offspring was exposed to literally at the 1 or 2 cell stage of development in the womb of the mother.
hmm.
And the link is alterations in the epigenome. So before that study was produced or published in 2003, no one ever used the word epigenetics to describe the mechanism for this type of thing, fetal origins of adult disease susceptibility. Afterwards, I don't think there's anybody anymore that doesn't use it, because it is the mechanism for the developmental origins or fetal origins of adult disease susceptibility. So we totally changed the way people in the general public and in science look at disease susceptibility. It doesn't happen just in adulthood. A lot of it happens back at the time of fertilization.
Did you guys ever experiment with giving those same methyl vitamins to the offspring and if that altered um, their expression at all after they were born?
No, we didn't do that, but the problem is you don't know what endpoints to look at. So now at least, as I said, once we had the human imprint home, that kind of study is in a way what we're gonna be doing in humans.
Mm,
Not only gonna look at the epigenome or these imprint control regions for their methylation in the offspring, the babies, but also in the mothers. And that'll be very interesting. I don't know what we'll find, but we now know places that are variable epigenetically, which are gonna cause changes in disease susceptibility and development. So there's nobody who's gonna argue that these are not important regulatory elements, and when they're out of whack, that's not good. So that's the studies that will come up now because we now have places to look, and they're not the only ones, Ashley. I mean, there's gonna be other ones, but it's a
mhm.
place to start where we can start doing organized studies to address the types of questions that you and your audience want answered. Is what if I do this, does it do anything?
Right.
Right?
Right. Yeah. That's the question.
Even aging. I mean, there are programs now to look at biological aging. I mean, if a person is exercising a heck versus ones that aren't or whatever, does it affect the I don't know. I guess there are people that are doing this type of stuff now, but that kind of stuff can be done now too,
Mhm.
because we have endpoints to look at. In effect, we have scales. We have molecular scales that we can stand on and we can say, yep, you weighed 150 here and now you weigh 170 and you're doing well, or you weigh 200 and you're not doing well. I mean, that's all you're doing is looking at methylation too high or too low. That's what you're going to be looking at. It's the same
Sure.
thing.
My question is so these studies were done, 20 plus years ago, and I feel like Western medicine hasn't caught up to that at all. Um, where do you see that? I mean, there's like you said, there's more and more research coming out, but it just seems like, from my standpoint and who I see that there's no real conversation in the doctor's office other than, here's a GLP or, for weight loss specifically, or here's this drug to lower your cholesterol when really if we change some lifestyle things, a lot of those markers could decrease by themselves and express differently. So how do you see that being implemented into, I guess, mainstream medicine?
Well, I'm gonna just use myself as an example. I told you I graduated. I retired from Duke in 2012. I weighed 260 pounds back then, and now I weigh about 178. So
Wow.
over that period of time, I lost weight, and this is way before they had the weight loss drugs. So how did I do it? I did it by changing my diet and by exercising. And all of the values, the cholesterol, and it wasn't because the doctor told me to do it. I said I was going in size 44 to 46 pants,
Mm,
and now we're 36. Yeah.
mm,
And
quite different.
I said to myself, "Enough of this. I'm done with this."
Mhm.
Everybody has to get to a point where they say, "I've had enough of this, and I gotta do something." So now in a way, I mean, I guess there's an easier way to do it with the weight loss drugs, but I did it the old-fashioned way, and in fact, even people that are using weight loss drugs, they need to change their diet in conjunction with that, because if they don't, if they ever got off of these drugs, they'd put all that weight right back on again because they're not eating in a healthy way. So, I see clinicians treat problems. That's what you do. Somebody comes in, you treat a problem, right? And that's the way medicine's set up. They're not set up so much to say, the reason you're there is you're 60 pounds overweight and you're putting all of this stress on your spine or whatever, that problems at your knees and that kind of things, and you gotta get that stress off. And we'll help you as much as we can, which I'm sure you do, and physicians do too, but you gotta help yourself, and the only way you can help yourself is by dropping the weight. And the only way you'll do that is changing your diet. and exercising. You have to do it in conjunction. Exercise by itself will not do it. I mean, you'd have to be exercising like crazy to
Mhm.
counteract, one little candy can do you'd have to exercise, as 30 minutes to get rid of the
Yeah.
candies in a half a Snickers bar or something like that.
Mhm.
It's not easy to do by exercising. The easiest way to do it, actually, I use easy because I've done it. It's not easy, is to not eat it.
Mhm. Do you think from an epigenetic standpoint, when you talked about the mice and I don't remember the gene name, but the gene for
Goody.
hunger,
Oh,
uh
the melanocortin-4 receptor is the
yeah,
CH
that one. That
yeah,
one.
that brain receptor, right.
Yeah,
Yeah,
that one.
yeah,
Um, that has a similar effect too, and it almost snowballs when our health gets out of control. It's like our epigenetic makeup is constantly ever evolving and ever changing, and so the more unhealthy we get, the more we're triggering those
yeah,
unhealthy habits, and so it probably takes more effort and more, um, in addition to willpower, but that, like, identity shift of, like, enough is enough for us to really make some changes later on, and once that snowball gets a little bit smaller, we have biology on our back now again, where when we're going down the road towards unhealthy, it's almost it's almost like a double-edged sword, like, it makes it even harder because that epigenetic makeup is not helping us out in that favor. Is that
yeah.
somewhat correct, or am I interpreting that?
No, I think that's true. I mean, from my own experience, it isn't easy, that's for sure. I'm not telling people this saying that this is an easy road. It's not an easy road. But, I was able to do it and, other people too, and some people have more difficulty. But it's the weight that really causes ultimately it's everything is out of whack basically, epigenetically, every way you can think about it probably, and everything starts coming in. It was amazing, all the things just start going down to where they should be when you get down closer and closer to your normal weight. I'm still not at my it's like, I think about building a house, the easiest thing is to frame the house. In other words, to lose the weight initially. 'Cause the first
Mhm.
weight you lose is water weight, so you say, "Yeah, boy, I'm going," but really not. It's because you're getting rid of sugar and you don't have as much water in your body, so you just are losing lots of weight. Then it gets a little harder, and then when you get down at the end, it's like finishing, like all the finishing work. It takes, like, months to do what you could have done in 3 or 4 days when you were just roughing it in. So I'm at the finishing level, and I'm not sure I'll ever get to the end of the finishing level before I'm not here anymore because it's even harder to do when you're down there because you're close, but not quite close enough yet. I would say that, according to the BMI, I am still right at the edge of being overweight. And getting it just a little bit more is not easy. So all of you out
Mhm.
there that are running into this I know what the problems are, and they're not easy. But it really is all of it involves basically ultimately reprogramming your epigenome. That's exercise does this too.
If you
What's
could
that?
give everyone one idea to take away from epigenetics or improve how they live, what would it be?
I think the main thing is that you're not only especially if you're younger like yourself, what you do is not only affecting you, but potentially the epigenome and its alterations is affecting your germ cells. So it's going to be affecting your future generations. And if you have a faulty program and it's passed forward and it's not being eliminated, it'll be passed forward for generation upon generation. This is literally the sins of the father, which was, obviously it's a very biblical thing, but this is a mechanism for this occurring. It just goes from generation to generation to generation with almost 100%. Uh, uh, what's the word? Penetration is the word they use in genetics. It's a very penetrant mutation that's occurring. The epi mutations, in a way, are more penetrant than many of the genomic mutations because if you have a single point mutation in the genome, if you inherit a good copy, you don't see any effect in the offspring. Whereas with
Right.
this, If you have a faulty program here, just try this sometime. Take a faulty program that doesn't work in this computer and put it in another computer, which would be your offspring. It doesn't work there either. Now take it and put it into the next generation. It doesn't work there either. So a faulty program, if it's not erased, when it goes through the egg and the sperm, will go on for generation upon generation before it's
Can these
ultimately cleared out.
can they be reversed after conception or once established, or are they fixed?
Well, that's one way of potentially if something is due, let's say there's an epigenetic change that, a lot of developmental Prader-Willi syndrome, Beckwith-Wiedemann, there's a lot of syndromes that we know that are due to imprinting deregulation. So one way of gene therapy for that, if you think about it, if you got one like this here, one copy from mom, one from dad, if you only have one copy and now that's mutated, you'll see a problem. So one way of going in there and changing that problem is through gene therapy, using CRISPRs and going in and trying to get rid of this and doing it genetically and get this thing back to not having this, but be like this, right? The other way of doing it is that you open up this copy here is probably perfectly functional, but it doesn't function because it's shut down epigenetically. So you have a mutation here. This is dead. And now you open this thing up epigenetically, and now you've got a functional copy and it works and you're just fine. So epigenetic therapy is different. You're trying to open up, in effect, these silenced alleles. And there are people working on this, and it's working. So they're just starting to do that type of work, but it's a completely different approach to, quote, gene therapy if your problem is epigenetic versus genetic.
What is epigenetic therapy?
Well, there maybe are compounds like valproic acid, which is being used, I think, is treating for epilepsy. It is an HDAC inhibitor. So it's been shown now to ameliorate some problems that are caused in imprinted genes where one copy is mutated and the other one's silenced. It opens up the silenced copy some, and you'll get some amelioration of problems, disorders and syndromes that occur because of imprinting. And I'm sure this kind of stuff happens in other places in the body. It's just that we have not been looking at it. And what'll happen is that companies, pharmaceutical companies, once the word gets more out that the epigenome is playing a role in diseases, that one potential way of treating these problems are going to be through drugs that alter the epigenome. And that companies would have to select for compounds that did that. You just screen for compounds that alter the epigenome.
Hmm.
Or you bring in CRISPR again to just do that, open up that copy in that location. These are things that'll be done in the future, but, the problem now, though, Ashley, is before you can do that, you have to know where the targets are.
Right.
And that's where we're at right now. So, people are, and I'm too, you get dissatisfied with the rate at which things are going. You wish they could go, go, go. And they are going, but they're not going as fast as most people, including myself, would like. But now we'll find targets, and then we'll start finding drugs that can open these up, and then that will start mushrooming out into a lot of other things that are broader or more constricted for certain specific syndromes that we know for their treatment. Yeah, that's where I see it going. It'll be called epigenetic therapy.
Interesting.
But it's not there yet. I mean, there are some compounds that actually already do it, and we don't even know it. As I said, valproic acid was used for the treatment maybe still is for treatment of epilepsy, but it's also HDAC inhibitor, which therefore it will alter the epigenome. So who the heck knows? I mean, part of the effect of treatment of epilepsy might already be epigenetic therapy. I don't know, but more than likely it probably is to a certain degree. That's how it works. And you
That's
didn't even know it.
interesting.
It was just a drug that was
Yeah.
used at work, right?
Right? It does
That's
its job, right?
you don't have to know
Interesting.
you don't have to know the mechanism by which it works.
Symptoms better.
So many of these drugs might already be affecting the epigenome, and they haven't been It hasn't been determined that that's the case yet.
Mhm. Yeah, that makes sense. Both from a positive and a negative way.
I wanna end up on something, though, that is very near and dear to my heart because it's in the area of radiation, and so I got into radiation biology out of coming through nuclear engineering. So the last study that we did at Duke was with, Autumn Bernal, who was a graduate student in my laboratory, and all the Agouti work that we did was done with chemicals, nutrition, non-genotoxic carcinogens like bisphenol A, that kind of stuff.
Mhm.
But what I was interested in is, could physical exposure to physical agents also alter the epigenome and therefore alter disease susceptibility?
Yeah.
So we did a study with low doses of radiation, the type of doses that you would get, particularly me as older, you get more of these, as you did CTs, scans, or, lung scans, those kinds of things. So a lung scan
Mhm.
would be, what, a point 0.1 centigrade. A CT scan would be around 1 to 3 centigrade, that in that area, very, very low radiation levels. To put that in perspective, if you were treated with radiation, you would be up to 200 centigrade per day over 30 days. We're talking orders of magnitude higher doses. These are very low doses of radiation,
Wow.
a little above background, but definitely above it. And so there is the risk assessment for radiation is called a linear no threshold model, which basically states that there's no dose of radiation that's safe. If you subtract background, the doses up here and effects up here will extrapolate through 0, 0. So at any dose, there's always some positive probability that you're gonna get a negative effect. So we did this study with this in mind, and with the agouti mouse, what you would be looking for is the bad. At any dose that you're looking at, you would find bad, and would even go worse as you got higher doses.
Mhm.
So we started at I think 1 centigrade, which is right at the bottom, I think, of CT scans, probably right where it is right now, and the dose that you would get if you got a CT scan. And we were expecting an increase in yellow animals because that is the unhealthy situation that you would see in this agouti mouse system. And that if we did a dose response curve, that we would see increases in the number of offspring that were yellow as we increased the dose. That was our hypothesis. I like simple. It's a good hypothesis. And that's what I was taught when I was in graduate school. So we did it, and I said we were starting to look at it, and I knew we were getting animals. The offspring were being born and all this kind of stuff, but Autumn wasn't telling me what was going on. I went into her office and said, "Autumn, we should be getting some offspring should be born. I mean, what's their coat colors? How are things going?" And this is her exact quote. She says, "It's freaky." Freaky is not a good word when you're in the middle of an experiment.
No.
Freaky is
No.
not good. It's freaky. I said, "What's freaky?" She said, "We are not seeing any yellow animals. They're all heavily mottled or completely brown."
Hmm.
So my answer, and I won't say it on, was, "Oh, you know what? We are now in the middle of the phenomena that's been referred to as hormesis," which I never believed in, "and we will now have to do a complete dose response curve. And even when you do that, you're gonna have great difficulty in getting this study published, because no one wants to think that a low level of ionizing radiation, which at high levels, I mean, is deadly, could be positively adaptive."
Hmm.
What we showed, and again, it's through changes in the epigenome. There's not just one system that's being exposed when you're exposed to chemicals or radiation. There's 2. One is the genome, and that's linear. Statistically, at no level will there be any there always will be a small probability that you're causing a negative effect and you're gonna get, let's say, cancer or whatever the endpoint you're looking at. But with 2 systems, the epigenome and the genome being affected, At low levels, you can induce the epigenome to counteract, and not only counteract, but actually cleanse the system to get rid of the pre-malignant cells so that in the end, you're actually healthier than if you didn't get the low dose of radiation. That's the phenomena of hormesis. It is correct, and it's same thing probably in chemicals as we see in radiation, but it brings in another point, Ashley. All your audience must know this. If a little is good and you get to the optimum, a lot is not going to be good. So the thing is with wine, you can say, well, a glass of wine might be good for you if you had it every day. But when I would say, but I can guarantee a gallon of wine a day is not good for you. And it's the same thing with any of these other things, including radiation, which is toxic up here, but at low doses, it's actually helpful. So you have to be careful. Why? Because it's a tendency, like folic acid and stuff you can buy by the bucket. Having too much methylation could be, it could be, I don't know, but it could be harmful. In other words, the level we were using in our study maybe was optimal and we showed that it was positive. But if you get up higher than that, my guess is you would find a dose where it's not optimal anymore, that it becomes toxic.
Mhm.
So it's not good just to pound a lot of stuff in you. If you do it by eating, for example, to bring your levels and stuff up, or you're careful, you're gonna be okay. But if you don't do that and you start eating too much of this stuff, you could get to a point where whatever you thought and was actually helpful at low doses becomes now harmful.
So you're telling me that I can eat a little bit of Doritos because it brings me joy and
Yeah,
it'll
yeah.
bring me way more health, but I just can't eat the whole
Actually,
bag.
no, I'm really in a way, I'm not actually being totally facetious, but it's hard to do that when you're talking about your food, isn't it?
No, I'm just joking with you, but, like,
No, no,
I
no, I am not joking.
Oh,
My guess
it
is pretty much everything is sort of like that. The problem
yeah,
is limiting yourself down to that low level, and the other way around, it's easier to go to the toxic level because it's good. I mean, the stuff tastes good,
right.
you eat too much.
Mhm.
Yeah.
Yeah,
So that's my end point. I got in the fact, that even physical agents, something you would never think would ever have any and, by the way, that positive adaptive effect could be blocked by eating antioxidants. Why? Because most of the radiation damage is done through indirect effects through the formation of free radicals. So
mm,
if you suck up those free radicals are important in ultimately altering the epigenome. If you suck those up, that messenger system is gone and you see no effect at all. So everything has to be done, even exercise, if you think about it. You
right.
exercise one day, you need to let your body have a rest. Why? Because if you keep doing that and doing that and doing that, ultimately there's too much free radical formation and you haven't let your body have a rest. And it becomes bad. You start
Mm,
going
mhm,
down. Whereas you exercise, you generate this, it makes this effect. It says, I got to build up your muscles and change the programming. Then you hit it again. And that's the way you ultimately build up your muscle and you get good health. Just going out and running yourself to death every day is not going to be good for you.
right.
And it's the same thing with everything. Moderation is probably, that's the last thing I'll say. My mom, I said when they say, "Well, what do you think is gonna happen?" I said, "Wouldn't it be interesting after all the billions of dollars we're gonna spend on this stuff looking at nutrition, its effect on the epigenome and health and all this, it comes down to what my mom said to me when I was little. You're gonna eat all those green things on your plate, or you're not getting up from the table. And secondly, you're not getting out of this house without eating breakfast." Those are the 2 ending things that she told me, and it's what I still do.
I think she has some merit and
I
wisdom
think
in
so.
there.
That's
Well,
mother advice.
right? It's intuitive.
Yeah.
Well, thank you so much, Randy. Your expertise, knowledge, and just profound understanding of epigenetics is it is such an honor to be able to talk to you today. And I'm just so grateful for this conversation. I learned a lot, and I know the listeners are going to be learning as well as they listen to it. So thank you
You're
very
welcome.
much
Thank you for inviting
for
me.
being here. I really appreciate it. And um, we'll be connected.
Okay, bye.
Thank you. Thanks for tuning in to Love Your Life and Yourself. I hope today's episode gave you one more tool or one more truth to help you thrive in your health, happiness, and resilience. Remember, loving yourself isn't selfish, it's leadership. And it's the foundation for building a life that actually feels like yours. If this spoke to you, hit that follow button, leave a review, and send it to a friend who needs to hear it. And if you want to dive even deeper, grab a copy of my #1 best-selling book, Love Your Life and Yourself. It's the heart behind this podcast, and it's helped thousands of women step into a life they actually love. You can find it at drashleyrath.com or through the link in the show notes. Until next time, take care of you and keep choosing to love your life and yourself.